Okay guys, so in this video I wanted to talk about the next three strata in your epidermis. So just want to start where we left off, the stratum spinosum. And we said that the keratinocytes in this stratum produce two things. Will begin to produce two things as they move through the spinosum. Lamellar bodies and keratohyalin granules. Okay. So what do these two things do? Well, as these keratinocytes move through the spinosum, right? So the spinosum is actually has a lot of, it's a thick layer of cells. I'm just only drawing one layer of cells for now. I'm not going to specify. I'm not going to talk about the layers of cells, how thick the layers are. But if you want, you can look it up. But anyways, as these keratinocytes move through the stratum spinosum, eventually they'll reach a layer called the stratum granulosum. Okay? And that is going to consist of keratinocytes that have lamellar bodies and keratohyalin granules. So they just started being produced in the spinosum, and now they are abundant in the granulosum. So I'll draw these little granules. Some granules here too. Okay, so stratum granulosum is going to have lamellar bodies and keratohyalin granules. What are these going to do? Well, the keratohyalin granules are going to contain two proteins, filagrin and tricohyalin. Okay, and these are both going to promote sort of the aggregation of tonofilaments. So tonofilaments and when they aggregate, when they sort of bundle together, they form tonofibrils. So I'll try and draw a bunch of them together in a way. Okay, so tonofibril. Okay, so tonofilaments form tonofibrils when they bundle together. Now, what is this going to do? Well, that's actually going to speed up a process, or it's going to result in a process called keratinization. So it's going to increase the process keratinization. So I'll write it up here, keratinization. Okay, keratinization. E. Okay, keratinization. And what keratinization is it's actually a cell, the process of a cell dying. So in the skin, so cell will actually die. So it won't have any of its organelles anymore. So I'll draw some of its organelles here. This is before keratinization takes place. Okay, it won't have any organelles left, and it will actually have a bunch of intermediate fibers, so like tonofilaments, forming sort of a scaffold inside it and taking up a large portion of it, almost entirely filling it up with this scaffold. Okay, and that is keratinization. So now in your stratum lucidum, going to have a bunch of these. Oh, I forgot to draw my desmosomes. These desmosomes need to be here. Yeah. So now you're going to have these cells. Still have the desmosomes. Okay. And cells in the stratum lucidum are either very late stage keratinized or they are keratinized. Yeah. So they're almost keratinized or they are keratinized. So that's the stratum lucidum. Now, so we've established that that is keratinization. And this actually helps form like a strong structure in the outer layer of your epidermis. So the outer strata in your epidermis. Now, what do lamellar bodies do? Well, I'll make some space over here. What lamellar bodies do? Well, I need to draw a diagram with. So this is an alive cell, okay, so probably in the granulosum. All right, and this would be any sort of dead cell or keratinized cell. Keratinized. So this is a keratinized cell. And your lamellar body is going to be inside your live cell, okay? It's going to contain contents that are actually going to contribute to the formation of an important layer in your skin. So this is your lamellar body. I'll just draw a line. So this is the lamellar body. And its contents are going to spill out into this area, okay? And they're actually going to contribute to forming part of this layer, part of a layer over here, or part of a barrier over here. And this barrier is called your epidermal water barrier. Okay, your epidermal water barrier. And it forms part of this epidermal water barrier. Now, what is your epidermal water barrier made of? It's made of two parts. A cell envelope and a lipid envelope. And your cell envelope is going to mainly consist of structural proteins. So those are proteins that are going to, provide structure to your skin, right? To wherever they are. And your lipid envelope is going to be made of mostly lipids. And your lamellar bodies actually contribute to that lipid envelope. So this area over here, so if this is the membrane, right? That's the membrane. Then all of this area, okay? So including all of this area that I'm shading in would be your lipid envelope. And this over here would be your cell envelope. That might not be the most accurate drawing of it, drawing with scale and everything, but it gives you the it gives you the idea of what it might look like, of sort of where it is. Okay, so that's the lipid envelope and that's the cell envelope. Okay, and that's what lamellar bodies do. Now, let's move on to the stratum corneum. So let's make some space actually. Whoa. Okay. Let's just erase this. Shoot. Okay. Um, just erase all of this. Yeah. Okay. Stratum lucidum. Yeah. And we have not erased the keratinization yet. Okay. So, some room. Top layer is going to be called your stratum corneum. Your stratum corneum is also going to have dead cells. But these dead cells are going to be sloughing off. Okay? How are they going to be sloughing off? So I'll draw one more. One sloughing off. Well, they're actually going to be sloughing off because their desmosomes are being degraded. And once their desmosomes are degraded, they are no longer attached to their fellow keratinocytes. So they can just slough off. pH is actually going to control whether or not these desmosomes are degraded or not. So there's a protein, so cali, that's not a K, okay. Kallikrein related serine peptidases are going to work to degrade these desmosomes. So they're going to degrade these desmosomes. So how is their degradation controlled? Because there might be cells underneath these that we don't want to degrade yet, right? So there might be cells underneath these that we don't want to degrade yet, right? Because they're not at the top. So why would they want to slough off? Because they're lower down. So they're actually going, the way to control this is actually by pH. So higher up in your corneum, you're going to have a lower pH. Over here, you're going to have a neutral pH, okay? So lower down, you're going to have a neutral pH. Okay? So when there's a lower pH, these proteins are going to start to degrade these desmosomes. Now, an inhibitor is actually going to prevent these proteins from degrading any desmosomes lower down. So it's called a lymphoepithelial casal type inhibitor. Yeah, so, oh, forgot the M. Lymphoepithelial casal type inhibitor. So, actually, let me just fix that right over there. Okay. What this is going to do is when it interacts with these proteins over here in a neutral pH, it's going to prevent any degradation. But in a lower pH, these proteins will actually be able to degrade these desmosomes over here. And these cells will be able to slough off, or they will slough off. So that pretty much concludes this video on all of this. On the other three strata in the epidermis, yeah. Just a reminder, just to recap, we found out what lamellar bodies do. They contribute to the epidermal water barrier. and these keratohyalin granules contribute to the to keratinization. Now, the epidermal water barrier forms a layer. So just to recap, we found out that these lamellar bodies in the stratum granulosum contribute to the epidermal water barrier, and the epidermal water barrier works to stop water from. It works with homeostasis, so it works to maintain homeostasis. And these keratohyalin granules are going to work for are going to contribute to keratinization, which results in these cells with sort of a scaffold inside them, so mainly tonofilaments inside them. And these are going to slough off in the stratum corneum once they're near enough to the surface. Something to remember is that the stratum lucidum is actually only present in thick skin, so not in thin skin. So thick skin, like the calluses on your feet. If there isn't any thick skin, it will be your stratum granulosum, and then it will go straight to your stratum corneum. Okay, so that pretty much concludes this video on the other strata of the skin. Thank you.